So let's review the structure of DNA. The simplest component of DNA is a nucleotide. More precisely, it's a deoxyribonucleotide. It has a backbone made of phosphate group attached to a 5' carbon of a sugar. That sugar is known as deoxyribose. These form the backbone of DNA, or you can think of them like the sides of a ladder. The code of DNA is determined by the nitrogenous bases, and these could be adenine, thymine, guanine, or cytosine. Each nucleotide is linked to another by what is known as a phosphodiester linkage. And DNA has a direction. DNA is read from a 5' to 3' direction. What this means is that DNA is much read like you would read a book. It starts with the 5' end of the strand. That is the strand that begins with the phosphate group. And then DNA is read towards the 3' direction, or toward the sugar end of the DNA. Not much was known about DNA until the 1950s. We knew that DNA was the code of life, but we had no idea how it worked. All we really knew is that it was made up of sugar, phosphate, and 4 nitrogenous bases. That's about it. Much of the foundational work of DNA was done by James Watson and Francis Crick. In 1953, Watson and Crick identified the structure of DNA. Throughout their collaboration, Watson and Crick proposed 3 characteristics of DNA. It was known that the 2 backbones of the DNA molecule line up side by side. However, they could theoretically line up in 2 different ways. They could line up in the same direction, that is, both sides of the ladder would be 5' to 3' in the same direction. Watson and Crick, however, proposed that the 2 strands of DNA were anti-parallel. This doesn't mean that they were perpendicular, rather it means that they line up side by side, but the direction of the DNA backbone goes in 2 different directions. And if this were to be true, Watson and Crick proposed that the pairing of the nitrogenous bases was complementary. That is, adenine bonds with thymine and guanine bonds with cytosine. If DNA were parallel rather than anti-parallel, it is theoretically possible that like nitrogenous bases would be paired with each other. That would be an A with an A and a C with a C and so on. But that's not what they proposed. Watson and Crick later shared a Nobel Prize for their discovery. Their discovery, however, depended heavily on the work of a single woman, chemist Rosalind Franklin, whose research was used without her knowledge or permission. Even worse, she wasn't even given credit for her work. She was not authored on her landmark publication identifying the structure of DNA. And it was Franklin's photograph of the DNA molecule that allowed them to see the structure of DNA. And this sparked a scientific revolution that was spearheaded by Watson and Crick. Watson said in regards to seeing this photograph for the first time, "My jaw fell open and my pulse began to race." The photo showed for the first time the essential structure of DNA, the double helix shape, and it also indicated its method for replication. She did not, however, know that these men were using her research upon which to base the article that appeared in the journal Nature. She wouldn't share the Nobel Prize either. However, this is probably not because Franklin was overlooked, but because she was dead. The award is never given out posthumously, and Franklin was diagnosed with ovarian cancer in 1956 at age 37 and died 2 years later, likely because of her exposure to radiation during her laboratory work that exposed the true nature of DNA. As scandalous as the situation with Rosalind Franklin was, James Watson and Francis Crick were truly revolutionary scientists. Watson and Crick proposed that the 2 strands of DNA served as templates or patterns for the production of new strands of DNA. In this way, DNA could be copied over and over and over again. Life requires this. They argue that these strands are copied according to a complementary base pairing. In other words, a DNA molecule opens up and the nitrogenous base pairs match up with their complement. A's with T's and C's with G's. Once the physical structure of DNA had been established, the next major feat was to understand how it replicated. And there are 3 possible ways DNA could replicate, and these are known as the alternate hypotheses for DNA replication. The hypothesis of semi-conservative replication predicts that the parental DNA separates and each strand serves as a template in which a new copy is made from each of the old strands. So in this picture, the pink strands represent the parental DNA strands, whereas the yellow strand represents the copied chromosomes, the daughter chromosomes. So in this picture, as the pink strands separate, a copy is made, and then each new double strand of DNA has a single parental strand and a single daughter strand. In conservative replication, the parental DNA is used as a template for the synthesis of a new molecule. In other words, both parental DNA strands are copied, and we would end up with the two strands of DNA, one with the original parental DNA and the other a complete copy of the parental double strand. The other possibility for DNA replication is known as dispersive replication. In this model, the new DNA molecules are a combination of segments of parental DNA and daughter DNA. So how is DNA actually replicated? We know based on the results of an ingenious experiment. In 1958, Matthew Meselson and Franklin Stahl designed an experiment that would determine which of these hypotheses would be supported. Nitrogen is a major constituent of DNA. Nitrogen-14 is by far the most abundant isotope of nitrogen, but DNA with a heavier isotope, nitrogen-15, is also functional. Meselson and Stahl grew E. coli in heavy nitrogen, nitrogen-15. What this did was create an E. coli strand with only nitrogen-15 within the DNA strands. After growing E. coli and nitrogen-15 for many generations, the heavy nitrogen E. coli was grown in a normal nitrogen medium, nitrogen-14. Then they followed the E. coli for a few generations and could determine the ratio of nitrogen-14 and nitrogen-15 in each generation. Comparing this ratio would allow researchers to determine which of these alternate hypotheses would be supported. Let's see why. So when the new generation of E. coli came of age, half of the DNA would be parental and half of the DNA would be a copy. Meselson and Wall could determine the amount of nitrogen-15 and nitrogen-14 and determine the percentage of parental versus daughter chromosomes. This would be true for all of the alternate hypotheses of DNA replication for the first generation. However, after 2 generations, these ratios would be different. Each of these 3 models makes a different prediction about the distribution of nitrogen-15 DNA in molecules formed after replication. The semi-conservative hypothesis predicts that each molecule after replication will contain one old and one new strand. This would mean that half of the second generation would be low-density DNA, those made entirely of nitrogen-14, and half would have an intermediate density of DNA. And an intermediate density of DNA would have half of nitrogen-14 and half of nitrogen-15. The conservative hypothesis predicts that each double strand of DNA makes an exact copy of itself each generation. If DNA were copied this way, the nitrogen-15 and nitrogen-14 ratio would be different. In the second generation, 1/4 of the DNA would be high density. That is, DNA would be made of 100% nitrogen-15 DNA, while 3/4 of the sample would be low density DNA. That is, made of only nitrogen-14 DNA. The dispersive model predicts that each strand of each new molecule will contain a mixture of old and new DNA. If DNA were copied via dispersive replication, each generation would consist of intermediate density DNA. That is, all the DNA would be made of parts of the nitrogen-15 and parts of the nitrogen-14 DNA. So what did they find? E. coli were grown for several generations in a medium with nitrogen-15. When DNA is extracted from these cells and centrifuged on a salt density gradient, the DNA separates out to the point which the density equals that of the salt solution. The DNA of the cells grown in the nitrogen-15 medium have a higher density than cells grown in a normal nitrogen-14 medium. After that, E. coli cells with only nitrogen-15 in their DNA were transferred to a nitrogen-14 medium and allowed to divide. DNA was extracted periodically and was compared to pure nitrogen-14 DNA and pure nitrogen-15 DNA. After one replication, the DNA was found to have close to the intermediate density, as expected. Since conservative replication would result in equal amounts of DNA of the higher or in lower densities, but no density of an intermediate density, conservative replication was automatically excluded. However, this result was consistent with both semi-conservative and dispersive replication. Semi-conservative replication would result in a double-stranded DNA with one strand of nitrogen-15 DNA and one strand of nitrogen-14 DNA, while dispersive replication would result in a double-stranded DNA with both strands having a mixture of nitrogen-15 and nitrogen-14, either of which would have appeared as DNA of an intermediate density. The authors continued to sample cells as replication continued. DNA from cells after 2 replications had been completed was found to consist of equal amounts of DNA with 2 different densities, one corresponding to the intermediate density of DNA of cells grown for only one division in a nitrogen-14 medium, the other corresponding to DNA from cells grown exclusively in a nitrogen-14 medium. This was inconsistent with the dispersive replication, which would have resulted in a single density, rather than the intermediate density of one generation cells, but still higher than cells grown only in a nitrogen-14 DNA medium, as the original nitrogen-15 DNA would have been split evenly among all DNA strands. The result was consistent with the semi-conservative replication hypothesis. Meselson and Stahl showed that each parental DNA strand is copied in its entirety, suggesting DNA is replicated via the semi-conservative hypothesis. However, they didn't give a mechanism. The discovery of DNA polymerase cleared the way for the mechanism of DNA synthesis. The discovery of the enzyme DNA polymerase led the way to understand more precisely the mechanism by which DNA is replicated. It was discovered that nucleotides are only added to the 3' end of the nucleic backbone. In this way, DNA is always synthesized from the 5' to 3' direction. In other words, DNA is read like a book, from front to back, never from back to front. DNA is replicated in replication bubbles in the chromosomes, and synthesis proceeds in 2 different directions. Since replication occurs only from the 5' to 3' direction, and since DNA is anti-parallel, synthesis occurs in both directions. Bacteria have a single origin of replications. In eukaryotes, DNA is replicated at several origins of replication. Let's look at the details of how DNA is replicated. DNA begins replication by being opened, unwound, and primed for replication. The DNA double helix is opened by an enzyme known as helicase. This is a specialized enzyme that breaks the bonds between the 2 strands of DNA. The DNA helix is then stabilized. This means that special proteins called single-strand DNA binding proteins attach to the open DNA strands in order to prevent them from automatically rebonding. Try to imagine unwinding a slinky into a string of metal with nothing but your bare hands. And you quickly notice that when you unwind the slinky, it would create some serious tension. So you might try to employ some tools to make the process of unwinding the slinky a little bit more easy. And that's what DNA does. DNA uses a special protein to combat this problem. This protein cuts and rejoins the DNA downstream of the replication fork, in effect relieving tension caused by the unwinding of the helix. In the last step of initiation, the DNA polymerase is primed. A special protein known as primase starts the process of replication by providing a 3' hydroxyl group that can bond with a nucleotide in order to form the first phosphodiester bond. Now DNA is ready to be replicated. The replisome is a complex molecular machine that carries out the replication of DNA. It's composed of 2 DNA polymerase complexes, one of which synthesizes the leading strand, while the other synthesizes the lagging strand. The leading strand is the strand of DNA being replicated continuously, and it's the strand that is being continuously polymerized toward the replication fork. All DNA synthesizes occur from 5' to 3', and in the leading strand, that happens continuously. However, the lagging strand grows in the direction opposite to the movement of the growing fork. It grows away from the replication fork and is synthesized discontinuously. It's called the lagging strand because it's synthesized away from the strand and lags behind the fork, slowing down the process. Because the strand is growing away from the replication fork, it must be replicated in fragments because the primase that adds the RNA primer has to wait until the fork opens to be able to add the primer. Once the primase attaches, the DNA polymerase begins to add bases to the 3' end of the primer and adds nucleotides away from the replication fork, producing segments of DNA. These fragments of DNA produced on the lagging strand are called Okazaki fragments. I have to say it like that. The orientation of these original DNA on the lagging strand prevents continual synthesis. As a result, replication of the lagging strand is a little more complicated than replication of the leading strand. This process repeats at the fork with further unwinding of the DNA. What is left is a bunch of Okazaki fragments along the DNA template. Finally, the DNA segments are attached to each other with DNA ligase. Okay, we've seen how DNA is opened, unwound, and primed, and how it's replicated in both the leading strand and the lagging strand. Now we'll find out how this process comes to an end. The end of the linear chromosomes in eukaryotic organisms are known as the telomeres. And at the end there's sort of a problem. When the whole primer is removed, it leaves a single strand section of DNA. This is eventually clipped off, shortening the chromosome. Let's see what it looked like. At the end of the telomere, the DNA polymerase continues along, business as usual, and slides off, completing the leading strand. Just like a zipper without a stopper, it just zips off. However, once the last Okazaki fragment is copied, however, it can't just unzip like the DNA polymerase of the leading strand. This leaves an unreplicated end. This will eventually get cut off. Interestingly, telomeres don't contain any genes. Rather, they're just short, repeating stretches of nucleotides. In order that the DNA molecule doesn't get smaller and smaller and eventually lose all functionality, these repeating nucleotides are copied onto the end of the lagging strands by a special enzyme known as telomerase. This prevents the lagging strand from getting shorter. DNA is incredibly accurate. For every billion base pairs that are copied, only a single base pair is copied wrong. However, even though DNA is this accurate, it has a built-in spell check mechanism. Special enzymes, known as repair enzymes, zip down the DNA and remove defective bases and repair them with their appropriate base pair. This molecule is known as DNA polymerase III, and this is how it works. Even though DNA is extremely efficient at copying itself, and it has a built-in spell check mechanism with the DNA polymerase III, it still makes mistakes, and these are known as mutation. Most are inconsequential, some are detrimental, but every so often a mutation comes around and actually provides an evolutionary advantage. And now you know the secret of life. You're welcome.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces. First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together. Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.